I'd like to welcome you to the second day of this great conference in this amazing venue. My name is Lara Schlafke. I'm a professor for medical engineering at the University of Applied Science in Dortmund, and I would like to welcome you to the first session of this morning where we talk about, what's the session's name? Novel special focus session about novel contrast in imaging. So it's not only about MRI, what you see today, what you hear today, and I'm happy that we have 3 invited talks following by selected oral presentations. And I'm happy to chair this session together with Hermine.
So, hi everyone, my name is Hermine Kan. I work at Leiden University Medical Center. And I want to also take like 1 minute to introduce this little glass house that you see here sitting in front of the stage. Because in the Netherlands we have the national radio station who's sponsoring a good cause every year. And then for that cause, 3 DJs are sitting in a glass house for a week, the week before Christmas. And they don't eat, they only drink. And the good cause this year is Spieren voor Spieren, which is an organization that supports research and care for muscle disease. So we as researchers thought it would be a very nice idea that we take the little glass house on tour. So we take donations for this and we will all give it to the good cause. So if you want to help, find me, or look at the QR code, which will also be on the screen later on. So with that being said, I would like to introduce our first speaker, which is Teresa Gerhalter, and she will share her information on opportunities and challenges with sodium imaging.
Okay, perfect. Good morning, everyone. So I'm delighted to kick off this session on novel contrasts in imaging with a focus on sodium MRI. So first, I also would like to place sodium MRI in the broad landscape of muscle MRI, showing here along with different aspects of muscle damage and degeneration. So, of course, we talked a lot already about yesterday, so we have the muscle degeneration where we have the muscle tissue replaced by fatty fibrotic tissue, which we can capture with Dixon-based methods as well as proton spectroscopy. And we also have other quantitative MR methods to capture different aspects. So we have T2, T1 mapping, and diffusion, where we focus on the inflammatory stage of muscle diseases, where we can depict inflammation, edema, necrosis, as well as microstructural integrity. And then on the left side, we also can focus on the functional disturbances of a cellular level, where we focus on disturbances with the ion homeostasis, metabolic stress, and mitochondrial dysfunction, by using different MR methods, such as sodium MRI and phosphorus spectroscopy. So why is the ion concentrations so important in muscle tissue? So in healthy tissues, we usually have a very tightly regulated extra and intracellular concentration of the sodium ions, which is kept up by the sodium-potassium pump as well as specific channels and an intact cell membrane. And this is very important for the cell viability and excitability as well as the metabolism. And in diseased muscle, this can be disturbed either through an impaired sodium-potassium pump, different impaired channels, membrane leakage, or early cell injury that could all lead to an increase in the intracellular ion concentration or changes in the cell volume due to osmotic imbalances. And this makes sodium MRI a promising tool to depict the ion homeostasis to actually focus on early pathological changes in muscle tissue. However, there are some challenges associated with sodium MRI. So sodium MRI, the sodium nuclei, has a fundamentally different set of MR properties compared to conventional proton imaging. So first of all, it has a spin of 3.5. That means we also have quadrupolar interactions that I will mention later on. We have a gyromagnetic ratio, which is about 4-fold lower than for proton MR, leading to also much lower NMR sensitivity. And on top of that, we also have a 1,000-fold lower in vivo concentration of sodium ions compared to proton. And taking this together, we really have a very weak sodium signal compared to proton MR, as you can see here. In proton MR, we have a general signal decay that decays with a T2 star, but then when we talk about spin of 3.5 nuclei, we also have quadrupolar interactions that lead to very short relaxation times. As you can see here in blood, we, for example, have a T1 for sodium of 50 milliseconds and a T2 of 24 milliseconds. And on top of that, in biological tissues where the sodium ions experience a restricted mobility, we have a bioexponential decay. So we have a T1 of 30 milliseconds. In theory, this is also a bioexponential decay, but we cannot really detect this bioexponential decay. And a T2, a short T2 of 3 milliseconds and a long T2 of 27 milliseconds. So there are some challenges, but we can try to address them by increasing the voxel sizes, increase the acquisition time, as well as use optimized pulse sequences with very short echo times to capture the fast signal decay. And then we can also try to use higher field strengths to boost our SNR. And what can we actually now detect or measure with sodium MRI? So usually when we have a spin density weighted sodium image, which is also referred to as TSC, apparent tissue sodium concentration, sodium content, or sodium concentration in literature, we have a look at all the ions. So it doesn't matter in which environment they are. But on top of that, we can also gain information about the molecular environment of sodium ions, based either on the relaxation time or the quadrupolar interactions. So as you can see here, you can use inversion recovery reparation pulses to suppress the signal from the liquid sodium ions. So you can see here that the blood vessels are more or less suppressed, and also like here the reference tubes that are only filled with like liquid sodium chloride, the signal is suppressed with the inversion recovery. And this has been in the past also referred to as intracellular sodium concentration, but I need to emphasize that this is not the case, and we should restrain from calling it intracellular sodium because we cannot distinguish intra from extracellular sodium solely based on the relaxation behavior. And then we also can make use of the quadrupolar interactions to get more information about the environment of the sodium ions. So we can use multi-quantum filtered, either like triple quantum filter or double quantum filter, to get an idea if the sodium ions are in an isotropic or anisotropic environment. And how does this work? So I just want to try to briefly elaborate on that. So proton spins have a spin of 1.5, so you have 2 Zeeman levels, and only like a single quantum coherence is possible. However, for sodium nuclei with a spin 3.5, we actually have 4 levels, energy levels that are possible. And as you can see here, in a liquid, we only have single quantum coherences. However, in like biological tissues, we in addition have like fluctuating quadrupolar interactions. So we have more transitions that are possible. So we have like double quantum coherences and triple quantum coherences. And we can, with dedicated multi-quantum filters, extract those signals as well. And so we can get an information about the nuclei that are restricted in their mobility using a triple quantum filter. And we can also use a dedicated sequence with like a double quantum filter using magic angle excitation. And then we get only the information from the nuclei that are restricted in their mobility in an anisotropic environment. And then we also have a rare case, for example, in cartilage, where we have like very ordered environments where we don't have fluctuating quadrupolar interactions, but static quadrupolar interactions, and that would also lead to a shift in the Zeeman levels. So let's move now to the clinical applications. The first sodium images were acquired in 2000 in myotonic dystrophy patients. And as you can see here, over the past years, some publications have been published not only in brain, but also in muscle. And from those publications, different muscle pathologies were investigated. So there have been muscle dystrophies, including myotonic dystrophy, Duchenne, Becker, FSHD, dysphalinopathy, and as well as some channelopathies. And I will just try to highlight a few of those publications. So here you can see an example from like a Duchenne study, where we have like a fat fraction map, water T2 map, as well as like a total sodium image. And in order to quantify the sodium concentration in the muscle tissue, we need to do some signal calibration using external reference tubes where we know the exact concentration that we put into that. Then we have a linear calibration curve, and then we can deduct the concentration within the muscle tissue. But of course, when we talk about muscle dystrophies, we also know that they have some fat replacement, so we also need to correct for the fat fraction in a later stage. And then we can have an apparent tissue sodium concentration maps for our muscles. The same can be also done for the inversion recovery preparation, that we can get a calibrated signal for the inversion recovery. And on the lower row, you can now see like corresponding maps for the Duchenne patient, where we can see some like slight increases in the fat fraction in the gastrocnemius muscle, bigger increases in water T2, and then we have like quite high increases in the total sodium concentration, as well as in the inversion recovery images. So we followed up those patients over a year, every 6 months, and here you can see the fat fraction depicted across the age, and the horizontal line is the line for the controls, and we see what we expect, that the fat fraction increases with the disease progression. And when we have to look at the same parameters for the water T2 and the sodium indices, we can see that we have a high increase in water T2 and sodium indices compared to the control values, and that this elevation is much higher for like the sodium indices, making sodium MRI a probably interesting tool to investigate early changes in muscle dystrophy. We then also had a look at FSHD patients and wanted to check the different sodium contrasts that I mentioned earlier, so inversion recovery as well as triple quantum filtration. And as you can see here, we have, of course, also acquired stir images to highlight the edematic changes, and it corresponds also with some of the sodium contrasts, but not always. So we also have, for example, here in the gastrocnemius medialis muscle, which appears normal in the stir images, increases in the, for example, TQF images. And yeah, we had a look at, in total, 19 FSHD patients, and we were also interested, like, how do the different sodium contrasts correlate with each other? So, which was very interesting to see is that the TQF and the inversion recovery, although there is, of course, a certain correlation, it is not a perfect correlation, so we seem to look at different mechanisms there. And we also subdivided the muscles according to the fat fraction, in low fat fraction, medium, and high fat fraction. And what we can see here is that especially muscles with higher fat fraction or medium fat fraction have increases in the sodium indices as well as in the water T2. And another group, so in Newcastle, they had a look at sodium MRI after exercise and dysphalinopathies. So they found that at rest we have increases in fat fraction, water T2, and total sodium concentration compared to controls. And then they did plantar flexion for some minutes and to see how the T2, water T2 and sodium would recover. And what you can see here is that we have the controls in light gray, that we have like increase in the water T2 and in the sodium, but we don't see really increases in the dysphalinopathy patients. And also no changes were observed in the relaxometry. And we are, of course, talking a lot about muscle, skeletal muscle tissue, but also the heart could be affected in neuromuscular patients. So there has been also like a setup developed in Erlangen and Heidelberg, where we could measure the sodium of the myocardium. And with dedicated respiratory and cardiac motion correction, we can here also monitor like the sodium signal in the heart. This has only been done yet in healthy volunteers, but could be a possible application also for NMT patients with cardiac involvement. So now I'm almost at the end. So we are of course, always trying to advance also the imaging accuracy in sodium imaging. So we try to attempt a spin density weighted acquisition. We also need to use external references because we don't have internal references as in brain imaging. And once we acquire our image, in the post-processing step, it's also good to have a look at different signal correction steps where we could even improve our accuracy. So we should always think about to correct for B1 and B0 and also partial volume effects. So for the partial volume effects, we actually have two effects we should consider. So we have spillover effects to do the signal smearing of the Weinspreckt function that should be corrected for. And then, as I mentioned earlier, if we have within a voxel like different tissue types, we should also try to correct for tissue fraction effects. And then, if we were not able to get a purely spin density weighted acquisition, we should definitely also correct for relaxation differences. And if there's motion, of course, also for motion. So, to conclude, sodium MRI still faces some challenges. We have a low gyromagnetic ratio, quadrupolar relaxation, and a low tissue concentration that result in a very weak signal, which requires fast imaging strategies, along with dedicated RF coils and multinuclear capability of the MR system, which unfortunately also comes with some additional costs. However, we then can also have some benefit because we can get additional information about the pathophysiological processes in the muscle tissue and information about the ion mobility and cellular integrity. And looking ahead, there has already been some interesting progress. So we can make use of multinuclear interleaving techniques to allow for a combined acquisition of the proton and sodium signal or the sodium and the potassium data. And new denoising and reconstruction methods could also further boost our SNR. And as the field also continues to mature, multi-center studies and standardization efforts will be also very crucial for translating sodium MRI to clinical applications. So with that, I thank you for the audience, and I'm happy to take some questions.
Thank you very much also for staying excellently on time. Kieran.
Thank you for a great talk. Just comparing the cases you looked at with the DMD study and the FSHD study. So in the DMD case, you're showing large elevations in sodium at an early time course. Were you able to get any feeling of the relationship between the sodium level and the T2 in terms of time? Did you see an evolution? Of one before the other?
Yeah, so we have to say like this were only done on 13 patients, so it's quite tricky to make some assumptions, but I think in general we just see higher sodium values, but of course this is also not only impacted by increases in the intracellular sodium, but also like enzymatic changes. So one would also expect we also seen a correlation between water, T2, and sodium, but we also see some muscles that haven't showed this correlation where we have normal water T2 values, but increased sodium signal. But I don't really have the data to really look at the time evolution.
And in the FSHD case, that was a cross-sectional study at one time point, I believe.
Yes.
But you were not, and you were able to make association between the level of fat and the sodium, but it didn't appear to be independent to that process. You couldn't see elevated sodium in the absence of fat.
Not really, no, not yet. I mean, we are currently running another FSHD study, and I have to evaluate the data because it's a longitudinal study, so that would be definitely interesting. But we know in FSHD it's quiescent for some time, and then suddenly we have an increase in disease activity and like a rapid replacement of the muscle tissue. So, and also from simulations that was, I forgot the group, but they carried out a simulations that the muscle tissue actually has a capability of maintaining for a while like increased sodium levels without being like really disrupted. So maybe there might be like, disturbances for a certain time, but we are not able to detect them because the cell can still handle it very well, and then suddenly we have bigger disturbances. Yeah.
Thank you.
Yeah, thanks for this really good talk. I have one question referring to the same data. So you showed that the sodium signal change was much larger than the Dixon, the fat fraction change. But what is the reproducibility of your method? Because you have to put these changes in context of reproducibility, which for Dixon is about 3–4%.
Mm-hmm. So of course it depends on all the correction methods that I also mentioned later on. So we also did, so usually for sodium we have a coefficient of variation of like 5 to 6%, which is not bad, but you could also, try to further improve it if you do like relaxation correction, B1, B0 corrections as well. And also what we found interesting is that we have quite nice coefficients of variations between a 3 and a 7 Tesla system if we include all those corrections, which speaks for 3 Tesla acquisitions.
Okay, thank you.
Thank you for your very nice talk. I have a question on the costs. Could you give us an idea of the costs of the coil? Because I understood you need a special coil. Mm-hmm. But also, where do you see the place of this kind of technique? Like for multicentral clinical trials, it seems difficult if the costs are high. Mm-hmm. So where do you put this technique? What is the place of this technique for the future?
So there are several manufacturers that are also building dedicated RF coils. So I only know like, it depends of course on your design of the coil, what you want, if it's only like a single tuned to sodium or you also want to have a double tuned like proton and sodium coil. So, but I would say roughly like 100,000 euros. So it's much less compared to what an MR system costs. And I think right now it's still in an investigation period, let's say. Like we have different centers across Europe that have like a sodium coil. Also, some people do just a home-built coil if they have like the expertise in-house. But I think we still need also like multi-center studies where we already use the coils that are in place to really prove that this is feasible, and that would maybe also encourage other centers to hop on the train.
Thank you, Teresa, very much. Thank you again. And I would now like to welcome Aurea Bach, and she's talking about muscle fibrosis imaging with MRI, the opportunities and challenges. I'm looking very much forward to your talk.
Thank you. Thank you for inviting me for this presentation. Okay, so we are talking about neuromuscular disorders, and this is the sort of histological image that we are used to see when we often see this kind of alterations that are combined. in patients with neuromuscular disorders, where we can see like cell degeneration, we can see fat infiltration, we can see inflammation, and increasing the extracellular matrix deposition, which we call fibrosis, all in the same place. Fortunately, we can assess those features, some of those features non-invasively with MRI and other methods. For example, we have very robust methods to assess fat fraction and disease activity, for example, with water T2. But we would love to have a third column on figures like this where we can quantify fibrosis, and unfortunately, we don't have yet a tool to do that. So I'll try to come with some ideas of what we could do. First, let's think about fibrosis. So fibrosis in neuromuscular disorders, or in general, let's take a step back. When you have a normal muscle and you have acute injury, we have first a peak on inflammation, and it leads to activation of fibroblasts, deposition of extracellular matrix, and then it reaches a maximum and starts to go down. And we start a remodeling phase where the cells, the extracellular matrix is remodeled and the cells start to maturate. The fiber cells that we generated start to maturate. What we see in histology is like this. We have first a normal muscle, then we have the acute injury there, lots of inflammation. We start with matrix deposition, and then we start the remodeling when we reach this stage here. And in the end, we have a completely regenerated muscle. When we have chronic injury, which is the case when you have dystrophies, we don't reach this peak. We always have some injury happening, and this means that we always have this inflammatory phase, and we have a constant accumulation of extracellular matrix, a constant stage of activated fibroblasts, and it leads to a gradual deposition of extracellular matrix in the tissue. So we start with a tissue that has been through a few rounds of degeneration and regeneration, and then it goes up to a point where the extracellular matrix is never remodeled. It starts to accumulate, and that's when we have fibrosis. Okay, that's fibrosis. This is an example of here a normal muscle, and here a model, a preclinical model of fibrosis where we inject botulism toxin, and we see these regions where we have increased extracellular matrix. So this is a model where we have just fibrosis. What do we expect to see in changes in this tissue? We expect to see changes in tissue microstructure. We see here the extracellular space is increased. We expect to see changes in the molecular composition of the tissue overall. We know that extracellular matrix is mainly composed by collagen, but other proteins as well, other components as well. So we expect to have an accumulation of these components. And we also expect to see altered function and mechanics of those muscles, indeed, because we are replacing contractile tissue by non-contractile tissue. Great. Can we assess any of those changes with imaging? And to try to answer this question, I'm going to break it down in a simpler question. First, can we assess those changes if we think about a tissue that has just fibrosis? And then, can we assess those changes in a more realistic scenario where we have fibrosis with inflammation, fat infiltration, dystrophy, or all the changes that we usually see in real world? So let's start with the simpler question. Here, I will go into details in this paper where we designed a mouse model with different levels of collagen accumulation or fibrosis by combining acute and chronic lesions in different genotypes and giving time for the inflammation to go down, but not enough time to have remodeling of the extracellular matrix. So we had fibrosis in different levels. And we started by we assessed various metrics. I'm going to talk about just three, the most interesting ones, I think. First, looking at the extracellular space. So we borrowed this technique that's usually used in cardiac MRI to assess fibrosis after myocardial infarction, for example, using T1 maps before and after injection of gadolinium contrast agents. And we did the same in the muscle. Sorry, I forgot to say that in our mouse model, we had one leg that had the injury protocol, so we had fibrosis in one leg, and the other leg was the control. So what we saw when we assessed the extracellular volume using MRI was that the injured leg had increased ECV, and it correlated with collagen content. So when we know that we have just fibrosis, or mainly fibrosis, ECV, as measured with MRI, is increased. The other thing that we did was to use a metric that is changed when we change the molecular composition, which is T2. So we know from imaging in general that tissues rich in T2, like tendon, they have low, rich in collagen, like tendon, they have low T2. So we measured T2 in the muscle of those mice, and we saw that indeed the injured muscle had lower T2, and it correlated negatively with collagen fraction. Great, so when we know that it's just fibrosis, T2 is reduced in the muscle. What about mechanics? So we used shear wave elastography. It's not MRI, it's ultrasound, but it could have been done with MRI. I will talk about it a little bit later. And we extracted this viscoelastic index, which is basically the shear modulus when the muscle is at rest and at maximal plantar flexion. And we saw that this viscoelastic index was increased in injured muscle as compared to control, and again, it correlated with collagen fraction. So when we know that it's just fibrosis, we can say that muscle stiffness is increased. Great. We have several metrics that assess that are sensitive to fibrosis, and we proved it. What about a situation where we have a combination of pathological features? So let's go back to that, the things that we think that fibrosis will change in the muscle. Now, if we think about dystrophies or neuromuscular disorders, we change from that easy and nice image to this more complex reality. We can have variable levels of fibrosis. So here in the mild case, we have just some accumulation of extracellular matrix, which might have already a big impact in function. But we can go up to this severe stage where we have a combination of everything. We have fat, we have inflammation, we have cell degeneration, we might have edema, and so on. Still, fibrosis will lead to the same changes that we mentioned before. So can we see how we could assess them in those complicated scenarios? Starting with tissue microstructure, there are several methods that are potentially or have been proven to be sensitive to fibrosis, like the ECV estimation that I showed in the mouse model, T1 maps, sodium, as Teresa was talking about, that can reflect changes in the extracellular volume. Susceptibility imaging, we know that when we change from intracellular space and we go through a membrane and go to extracellular matrix, you might have changes in susceptibility. It has been shown more like 25 years ago that if you have resolution high enough, you can even see the epimedian layers in the muscle. So could you use them to assess fibrosis in the real world? So I have an example here where the authors did calculate the extracellular volume in a similar way as I did for the mouse model, as I showed for the mouse model, and they observed in Becker patients now. And they observed here that in Becker patients, both in muscles with low and high fat fraction, there was an increase in the extracellular volume. And they took care to use water T1 for the calculations here, so it was not contaminated with fat. That's great. So ECV is increased in Becker patients. This could reflect fibrosis, but we know that in Becker patients, there is not just fibrosis. So how can we say that it's just fibrosis? When you have dystrophy, you have cell lesions, membrane lesions. You have edema. Edema will lead to an increase in the extracellular volume as well. So the extracellular volume that you measured, if you have edema, will be right, but it's not going to reflect fibrosis. We also have membrane lesions, so the contrast agent that we assume would be distributed only in the extracellular space can go now into the cells. So the extracellular volume that we're measuring is actually an apparent extracellular volume, not a real one. So there are some limitations when we try to assess tissue microstructure with MRI. And the big challenges are specificity. We can't really separate what's caused by fibrosis from the effects of inflammation, necrosis, membrane lesions. And also another big one is resolution. If you think about that image that I showed before with high resolution where we could see the epimedian, we can think that what is here inside those boxes is more or less what would be inside the voxel of this very high resolution image. And if you see here the quantification of connective tissue in these voxels, let's call voxels, you see that when you go from controls to mild or even moderate fibrosis, the amount of connective tissue inside the voxel is really small. even though you might have a very important impact in the function of those muscles. So we need high resolution and we need high sensitivity to detect those small changes with the voxel sizes that we have. Okay, what about molecular composition? So, as I mentioned before, we know that tendons have low T2, they have higher amount of collagen and low T2. We see the same in scar tissue. So here, when we have acute injury in the muscle, we have increased T2, or signal in T2 weighted images, and we have low signal in T2 weighted images when we have the scar established there. This is not published, it's a contribution from our colleagues from St. Petersburg, but even in patients with dystrophy, in this case dysferlinopathy, we can see if we can see cases where we see low signal in T1 weighted images and in T2 weighted images after fat suppression. This couldn't be disease activity. We know that it leads to high T2 when we have edema or inflammation. So we consider that it probably is caused by dense fibrosis, dense enough that it is possible to see it. But that's not usually the case. Usually fibrosis is not so dense, it's more distributed. There are other approaches that we could use to try to see fibrosis in those cases. This is another preclinical study where a mouse model of congenital muscular dystrophy type 1A was studied. We know that these mice, they have fibrosis, but in mice you usually don't have fat infiltration, right? So I'm going to talk about global T2 here. The authors measured T2 in these mice and they saw that, well, T2 is increased, reflecting the dystrophic lesions that the mice have. We usually see this in dystrophic mice. But if they excluded the voxels when calculating T2, if they excluded the voxels that had T2 higher than the average T2 in normal muscle, what they saw is actually that the global T2 in that muscle was reduced, possibly reflecting fibrosis. So if you exclude the voxels that are dominated by inflammation, you might see fibrosis. And when they treated those mice, they had normalized the T2. They treated for fibrosis. So very interesting, but still we might have combined the facts that they found a way to try to separate inflammation from fibrosis. A more elegant way would be to work with ultra-short echo time sequences where we can actually see the short T2 fraction, the signal from species with short T2. This has been shown to very reliably quantify collagen in phantoms. And it has been used to quantify a short T2 fraction in patients, which is very promising. But we still don't know if this short T2 could be affected by other macromolecules which also have short T2. If you think about actin and myosin, for example, they have short T2. What else could be changing the short T2 fraction apart from collagen? We don't know. There are some promising avenues there. Double quantum filtration combined with MT, T1 row might be more specific than this. I will not manage to go over this today, maybe on another talk, but we are not there yet. We don't have specificity yet. So we come back with more or less the same challenges that we had before. If we try to assess tissue composition or molecular composition of fibrotic muscle, we still have challenges with specificity. We have contribution of other pathological changes to the signal. We have fat infiltration can complicate a lot the modeling of our signal, for example, and we again have an issue with resolution. Okay, there is a possibility there, which is to use contrast agents that are targeted to extracellular matrix components. There are quite a few contrast agents linked to gadolinium that are targeted to collagen. For example, this EP3533, it has been used in several tissues. It has been shown increased signal in dystrophic muscle in mice. We have probes for collagen 3 as well, which is great because collagen 3 usually accumulates before collagen 1. So it's a nice way of trying to see the dynamics of fibrosis accumulation, remodeling, and so on. We have probes for elastin as well. So we could try to think about probes that would be very specific to proteins that are accumulating there when we have fibrosis. This is a great solution to increase our specificity, and they are usually quite sensitive, but we have a big question marker there is, can we translate this to clinics? They are at the moment all only used in preclinical studies, not in humans. Okay, what about mechanics in the muscle? There has been there were a few studies on mechanical properties in the muscle. I'm going to show this one with MR elastography in Duchenne patients, where it was observed that at rest, the dystrophic muscle has higher stiffness here, the light gray, as compared to control muscle. But when we have a muscle contraction here, in those cases, the stiffness in the Duchenne patients is lower. So this increasing stiffness at rest might reflect fibrosis, but this reduced stiffness there, when you have contraction, probably reflects the lack of the loss of contractile tissue. So that stiffness that we have when we contract the muscle, they won't have just because they don't have as much contractile tissue anymore. So again, we have issues with specificity. We are going to combine effects of lots of alterations that are happening at the same time, and potentially of resolution, because if you could have a high enough resolution, we could potentially see differences in the effect of where you have predominantly one alteration versus the other. Okay, not very reassuring, but if we try to summarize this, we could come up with tables like this, where we try to identify how each MR modality will be affected by fat, by inflammation, dystrophic lesions, and fibrosis. And if you concentrate just on fibrosis, it's quite hard to say, okay, I'm going to use this method to assess fibrosis with MRI. But we could try to think, coming back to Andrew Bloy-Mary's talks from yesterday, we could try to think about multivariate approaches, where we combine contrasts to try to separate what comes from fat, what comes from inflammation, what comes from fibrosis. Okay, what's there beyond MRI? So I'm going to show two examples very quickly. With PET, first, I showed this image before, where in the normal case and the dystrophic case, we have always these FAPs, the fibroblast activating proteins. They go up and after lesion, and they can go down if you have remodeling, or they can still be up when you have fibrosis. Well, we have fibroblast activated protein inhibitors that are very specific, that are going to be very specifically binding fibroblasts that are activated and depositing extracellular matrix. We could label them, and it has been done in the context of oncology. So this is the stuff that is used in oncology for already in clinics for diagnosis and for therapy. We could bind those FAPI, the fibroblast activation protein inhibitors, to a radionuclide, and then trace them with PET. So this has been shown to reflect regions with fibrosis in the heart already. This is great because we have high sensitivity, specificity. It has been already used in clinical studies, but it comes with the limitations of PET. Cost and use of radionuclides and availability and so on. What else? We have MSOT, which is multispectral optoacoustic tomography, which Lina is going to talk after me about it. But it's also a very promising technique, which is able to separate the contributions of collagen from other contributions in the muscle. It has been used in Duchenne patients already, so it is translatable, it has been translated already, but it comes with a big limitation, which is the depth. It's sensitive to, it can image tissue up to 3.5 centimeters depth. So not something that could be used for whole organ or even for adult patients. Okay, so wrapping up, my conclusions are, well, NMR imaging or spectroscopy is sensitive to fibrosis, but currently we don't have methods that are really specific to fibrosis considering MR. There are promising avenues to separate the signal from fibrosis, inflammation, fat infiltration, and so on. First, the multivariate approach, and also combining possibly other imaging techniques. And of course, this has a very important impact. It's something that should be continue to be studied because it helps understand the natural history in those diseases. It helps to identify potential therapy targets and to follow up therapies as well. Okay, that's it. Thank you for the attention, and thank you for my mentors, my colleagues, and funding agencies, and happy to take questions if we had time.
Thank you all very much, Kieran.
Hi, great talk. In those nice experiments early on, when you were looking at the animal model that you were either expecting or demonstrating to only have fibrosis, can you remind us of the dynamic range in the ECV you were measuring, and what's the repeatability in that kind of measurement on a preclinical muscle?
Well, we didn't really assess repeatability because we didn't repeat the scans in the animals. We just had one scan and then histology afterwards. So we didn't have time to, we should have done this. The dynamic scale, you mean the amount of collagen that we cover, right? Like how much fibrosis we have. I will show you, not because I don't remember by heart. Come. Here. Okay, so we go from collagen content that is from below 10% to up to 20-something%. That's the dynamic range that we have for collagen content. And for ECV, the range is from 5 to 12%. So it's very low. We could say that it's quite sensitive.
It would be quite fascinating to know how much of that is, uncontrolled biological variation versus, the inherent signal.
I think that we could say
variation.
that the biological variation we could assess by the control muscle. It's the same animals, so even if we have a systemic effect, it's going to be taken on the other leg, the leg that was not injured. And then we knew that when we did the model, we used two different genetic backgrounds that have different susceptibility to develop fibrosis. So even in a non-injured muscle, we might have more fibrosis in one background than the other. So this would be captured by the controlled muscle. So we still have quite a broad range here in the control, but it's higher in the injured muscle.
Okay, so there's still quite a, as you say, a really wide range of variability in that control muscle. Do you think there's anything you could do to? Is there anything in technique you could do to change that, or do you think that is absolutely inherent?
We could, when we think about the protocol that we used to induce fibrosis, we can see here, for example, this is one of the genetic backgrounds, the control, and this is one of the other genetic backgrounds, the injured muscle. You see, it's quite similar. We could try to have a different injury protocol where you have either more rounds of injury or in one genetic background or even the two, but in a way that we could cover a broader range, a broader dynamic range for collagen content, for example. Yeah.
Thank you.
One very short question. Go ahead.
Thank you for the talk. Do you think DTI could help to differentiate the fibrosis and the fat placement? So most of the time they coexist, but with the DTI, maybe reduced fractional anisotropy and increased mean diffusivity or increased radiation diffusivity suggest the fibrosis rather than only fat only.
So diffusion is one of the methods that we know is sensitive to changes in the extracellular volume, but it's also going to be affected when you have membrane lesions, when you have differences in cell diameter, when you have inflammation that will change extracellular volume as well. So at the moment, I don't know how we could model diffusion signal in a way that we could reflect this is just variation in the diameter, because FA, if you have low FA, it could be just because you have same diameter cells and a lot of extracellular space, or just because you have a lot of disorganization of the tissue, or you have larger cells. So at the moment, I don't know about a model that could disentangle them. I might be wrong, but I don't know how to fix it.
Thank you. So for the interest of time, we're going to switch to the next speaker. But thank you, Oria, again. So the next speaker is Lina Tan, and she will talk to us about optoacoustic imaging and muscle imaging.
Introduction, and also thank you, Aurea, for the introduction to optoacoustic imaging, which is an imaging modality I want to introduce to you too, because I believe maybe it's quite promising in the field of neuromuscular diseases. So this is my declaration of interest. So I work in the pediatrics department in Erlangen, and as a clinician, I meet the patients somewhere along their clinical course. So either, or usually, they come with disease manifestation already, but especially in our pediatric cohorts, sometimes and oftentimes we are able to actually give the diagnosis before the patients become symptomatic. And this is quite relevant because early detection also means that I might be able to early treat the patients. Now, this is interesting for causal treatment, where some neuromuscular diseases offer that, which is helpful because I can then prevent irreversible muscle damage or degeneration. And it's also important because symptomatic treatment also improves morbidity. And with the evolving field of medicine, I both have better detection of diseases and also better treatment with emerging gene therapies. However, we may never forget that longitudinal monitoring of these patients is relevant too, and with increasing diagnosis values and increasing treatment options, this raises the diagnostic need of longitudinal biomarkers that are able and sensitive enough to detect changes. And I believe yesterday we have had great talks and discussions about the fact whether our current imaging modalities offer this advantage. And me as a clinician working in the field of pediatrics, I'm kind of limited to ultrasound and MRI, and in very rare cases I do CT and so on. And we luckily have good data on MRI in neuromuscular diseases, and this is just an example from Professor Diaz-Manera, where they did mapping of muscle group involvement in late onset Pompe disease. And this is an example I chose because Anne gave a great presentation on Pompe disease yesterday. And also, we can see that there are specific muscle group involvements in these patients. So for LOPD, we see there's a trunk involvement, whereas in peripheral muscles, for example, the biceps muscles, we rarely see any involvement of the muscle tissue. And this is something we have looked at in our study, where later I'll show you the results for optoacoustic imaging, but we compared the imaging methods to routinely available methods, being the ultrasound and MRI in in-phase and in fat phase. And we can see that in moderately affected patients, we can see a kind of loss in muscle volume. However, the muscle structure seems to be integral. And when we look at severely affected patients, we can see there's a fat infiltration in these patients. And when we quantify the results, we're actually observing that there's no differences detected between healthy and diseased muscles, raising the question whether there is no muscle involvement in the biceps muscle or whether we are just not able to detect it early enough. And in Anne's presentation yesterday, she said beautifully that we need to have a biomarker targeting the patients who are mildly and moderately affected, especially since there's a gene therapy available for IOPD, and there was a paper published this year in June 2025. So this leads me to optoacoustic imaging, and we call it a multispectral optoacoustic tomography too, because usually in all the devices, they use multiple wavelengths. And before I go into the technical details of this imaging modality, I brought you a video, and so don't worry, these are my colleagues. Nobody came to harm for this video. So you see this is actually an imaging modality very similar to the ultrasound, so it's non-invasive, it's bedside applicable, and it's also providing real-time and quick visualization. And you see we send in pulse laser light. I will go into detail later. And we can see a real-time visualization of the optoacoustic visualization of the muscle. And we can also do a quantification on this, by the way. So we send in laser light, pulsed laser light in the infrared spectrum, so around 650 nanometers to 1,200, 1,300 nanometers. This leads to thermoelastic expansion, which let me show you. So this is an example for hemoglobin, for example, and this generates ultrasound waves which are then detected by the probe. And this has a great advantage since it's a light-based imaging modality, being that proteins in our body react differently to light. So in this image, I try to show you the absorption spectrum of different modalities, being, for example, oxygenated hemoglobin, but also deoxygenated hemoglobin, who react differently in the lower wavelengths. And when we look at collagen and water, we see there's an increase with signal wavelengths. So this, with the knowledge of how an endogenous chromophore reacts to light, we can visualize it without the use for contrast agents. And we are actually able to, in real time, non-invasively characterize tissue composition and even oxygen saturation. And one can even argue this kind of is a muscle function imaging modality too, if it reliably visualizes oxygen saturation. So let's go back to, I highlighted the 800 nanometers because you will see it quite often in this presentation. Why? And maybe some of you have already detected this cross point of hemoglobin oxygenated and deoxygenated. This is what we call the isobastic point of hemoglobin. So we believe at this single wavelength, we can best detect hemoglobin and therefore, for example, inflammation, but of course also a perfused muscle. So let's go back to Pompe disease. I quite teased you up to this point. Again, you've seen the MRI images already, and now you can see the 800 nanometer signal for the muscle. So again, this is the bicep muscle, and then you can see the collagen and the lipid. So we can see there's actually a homogeneous loss of muscle signal and an increase in collagen and lipid signal, but only in the severely affected patients too. And Hermine yesterday asked this question, so I spontaneously decided to add in this slide, because when we compare the spectrum of Pompe patients compared to healthy volunteers, we see there's actually an increase in signal, and this actually surprised us a bit because we were actually expecting less signal, and we were investigating why this was the case. And Hermine yesterday asked, are we able to have an imaging modality to visualize water accumulation? And this is exactly what we found in our preclinical Phantom study. So we used minced meat, added some glycogen, changed the concentration of the glycogen, and measured it with the same clinical device. And we see there's an increase along the signal intensity axis, being the y-axis, but no change in the spectrum itself. And actually, the spectrum is exactly like the water spectrum we see. So we are able to say we can't actually detect glycogen with optoacoustic imaging for Pompe disease, which we hope for, to be fair, but we can see there's increased water signal because glycogen just has a high water binding capacity. And when we now look at the unmixed parameters being collagen and lipid, we can now actually say that we are able to distinguish between healthy and diseased muscle in patients with high statistical significance. And when we compare to disease severity, and this is also something we've discussed yesterday, we can actually see that with increasing disease severity, we lose collagen signal, whereas we gain lipid signal. And somebody yesterday said this is exactly what we detect in neuromuscular diseases. So in the early stages, we see a remodeling, a more pronounced fibrosis, compared to in later stages where we see the fatty degeneration of the muscle itself. So just a quick summary. We have accelerated diagnosis and novel therapeutic approaches, and this demands sensitive biomarkers to monitor patients and their therapy response. And optoacoustic imaging as a light-based imaging approach enables visualization of endogenous chromophores and detects muscle signal differences consistent with pathophysiology of LOPD. And this is a study we did in 2022 where we examined the standardization and reproducibility of MSOT, and we see there's a good temporal and good intrarater and interrater reproducibility. So now with the knowledge we have, I want to transfer the knowledge to other neuromuscular diseases. And I want to start, because I'm a clinician, with some videos where you see the patients we're looking at. So you can see this is a pediatric patient cohort, and you can see all these patients, they have a proximal muscle weakness. And actually on the left-hand side, you see a patient with Duchenne muscle dystrophy, and on the left-hand side, you see a patient with spinal muscular atrophy. So two neuromuscular diseases with completely different etiologies and pathophysiologies. So for DMD, the dysfunctional or missing dystrophin leads to fibrotic or primary fibrotic muscle degeneration, whereas in SMA, this leads to a secondary muscle atrophy due to the degeneration of the motor neurons. And when we look at the optoacoustic image, so respectively, you see on the left-hand side the healthy controls, and on the right-hand side the deceased patients. You see for DMD, there's actually a homogeneous fibrotic degeneration, whereas in SMA you see a patchy pattern. So there are patches of lipids, there are patches of collagen, and so on. So the authors call it an inhomogeneous moth-eaten pattern. And again, we can quantify these results and we can differentiate between healthy and diseased muscle for DMD in regards of the collagen signal. And this also correlated strongly with the muscle biopsies. And for SMA, interestingly, the quantification also reflected the visualization we see being reduced mean signal values, but increased maximum signals reflecting the inhomogeneous patches we've seen earlier. We were also able to do 3D images in spinal muscular atrophy with a 3D probe, and we were able to detect that MSOT recognizes differences based on disease severity. And when we compare it to whether they are treated or not, we were able to see that treated patients actually show better signal values, which were, by the way, also comparable to the ones of healthy volunteers. Now I want to show the very last muscle study we've done recently in May this year. So I showed you that the imaging device is mobile, and so we decided to do a clinical study to go. This is what I call it. And we moved it from Erlangen to Frankfurt into a wellness hotel, which was quite nice. Also, especially for the imaging device, it had itself hotel room, so it was climatized. It was great for the imaging device itself. And we were able to detect or image patients with central nuclear myopathy, so a very, very rare and heterogeneous myopathy. And I brought you some images so that you can get a feel of how the patients present themselves and how the biopsy looks. And now you know why the diagnosis is called central nuclear myopathy, because the nucleus is in the center of the muscle. But actually, there are quite a few mutations leading to this histological result. But the problem is that they all follow a different inheritance, so the patients are highly heterogeneous. And with the next few slides, I want to just look at the patients with a ranitidine 1 mutation, because they follow an autosomal recessive inheritance, which is interesting because some of the patients actually have variants of unknown significance, and they would like to know whether they fall into the, let's say, symptomatic cohort or not. So this is some preliminary data we have of this patient group. And again, you see on the left-hand side, the reflected ultrasound image, then you see the 800 nanometer signal added on the ultrasound image, and then you see the oxygenated hemoglobin signal, and you see, and this is not surprising, that we lose 800 nanometer signal with increasing disease severity, or at least in patients with, who are homozygotes. So when we quantify these parameters, so for both a single wavelength and both unmixed parameters, we can actually detect significantly differences between healthy and homozygote and heterozygote and homozygote. And the differences, they can be detected in the muscle signal, muscle oxygenation, and the fat signal. And when we do a PCA analysis, this was something which did not surprise us, because the heterozygotes patients were in between the healthy and the homozygotes ones. And the next step, we want to include the mutation type and class, of course. So now we've discussed a lot about rare diseases, and now have a look at more common diseases, especially in Germany due to lifestyle, which are vascular diseases. This is just a quick tour into this topic. Now we have already volumetric OAI studies on vessels itself, but we know that peripheral arterial diseases actually lead to end organ damage, but there are rarely studies done on the muscle itself, at least in the field of optoacoustic. So this is what we did. We see, of course, there's not surprisingly a decrease in 800 nanometer signal. And when we compare the patients regarding their disease severity, and we differentiate them between intermittent claudication, so up to Fontaine class 2B, or critical limb ischemia, we see we can detect differences between the healthy ones and intermittent claudication and the healthy ones compared to the chronic ones. However, somehow we are not able to differentiate between intermittent claudication and cortical limb ischemia. So we were wondering why this was the case. Again, are we not sensitive enough, or maybe our protocol is not the right one? So someone came up with the idea, and this is also something we've discussed yesterday, about looking beyond muscle structure and more into muscle function. So colleagues decided to make the patients do some heel raising and then detect the gastrocnemial muscle. And in healthy probants, you see there's an increase in signal, whereas in patients with intermittent claudication, there's no increase in the signal. And when you quantify this again, now you can differentiate between those. So now just a quick outlook into some of the limitations we have with this imaging technology. So this is light-based, as I said. So our biggest challenge is actually penetration depth, and Aurea already showed 3.5 centimeters is around the maximum we can get, but when we are realistic, we only get realistic signal values up to the point of around 2 centimeters. So this works quite well in pediatric cohorts. However, in adults, this is quite limited too. So there's one approach where you improve the hardware, and there's already some work done there, but also there's the idea of implementing influence correction, where we take this into account and change the signal based on the knowledge we have. And there's just a quick overview. So first, when you want to do influence correction, you first have to segment the muscle, then you tell an algorithm to calculate the changes made by the muscle itself, and then you add the corrected image onto the original map. And we've seen by this we can increase the signal also in lower muscles or deeper lying muscles, or for example the vastus lateral. And also we can see changes in an increase in signal for unmixed parameters. And this is actually quite interesting for this central nuclear patient cohort, because we can see there's a stronger increase in the, lipid spectrum, whereas there's a less increase in the hemoglobin spectrum. And you've seen the ultrasound image for central nuclear myopathy already, so this is something we believe would help us in differentiating between healthy and diseased patients. So also next outlook, we are trying to implement multimodal imaging and also explainable AI. So this is a class activation map. And this class activation map tells you what the neural network used to differentiate and to classify the muscles. So this is a DMD muscle biopsy. And you can see that the neural network, so everything that is more yellow is more activation, uses the myosin structure and not the collagen structure. And every time the neural network used the collagen structure as reference, it did a wrong classification. And so I hope I'm still in time, and I'm happy to take your question. So thank you very much.
Thank you, Lina, and you stayed in time, so that's very good. Are there any questions? Jordi.
Yeah, thank you very much. That was very nice. So with this technique, how sure you are that you can accurately follow up the patients longitudinally?
Yes, that's a very good question, and something we actually need to look further into. So most of the studies we did were cross-sectional, to be fair, and we tried to implement more longitudinal studies. So we had studies where we, sorry, I just changed because otherwise it's still moving. So we have had studies on standardization and reproducibility. And so we have already had studies where we looked into whether examiners who are not trained are able to get similar results to the ones who are trained in optoacoustic imaging. And we've seen when untrained examiners get like a video-guided training or teaching, they can get similar examination results as examined or experienced examiners. So I think from the technical side, it's able to do longitudinal follow-up. However, we have to keep in mind that for laser, it quite fluctuates, so we need a better improvement on the hardware itself.
Yeah, but if we take into account that if we look at fat fraction,
Yes.
and we see that in some diseases like Pompe disease, there is an increase of 1% or 1.7%.
Yes.
Do you think that you can get that with the echo? I mean, I doubt it. I doubt that you can really draw a region of interest in the echo and see these different, just a 1% increase in collagen. Have you demonstrated that?
We have not demonstrated that, but that's due to missing longitudinal data we have so far. We have preclinical data on this, where we've observed in Duchenne pigs how, like when they were born with Duchenne, how the collagen signal changes within time, and we followed them up for 4 weeks, which is approximately the time where in pigs we believe there's the most fibrotic changes, and where we were able to see that with optoacoustic we are actually able to detect changes, where pigs with Duchenne muscular dystrophy showed increase in fibrosis, which was measurable, whereas wild-type pigs showed a plateau.
Thank you. There was a question.
Yeah. Thank you. Very impressive technique. Maybe to follow up on that, I noticed that on many of your y-axis has arbitrary units. How do you actually normalize or quantify that signal and compare it between subjects and groups?
Yes, this is actually a very, very good question, because this is one of the post-processing limitations we regularly face. So in general, you can't, or we don't try to, because we don't believe we measured the same, or maybe the laser intensity wasn't the same. So we're happy to do, this is why we always include healthy controls, because we need to see, okay, is there actually a change? But we can't right now differentiate between, let's say, SMA, DMD, LOPD, and so on. But these are future plans we want to work on. So thank you for raising up this question.
More questions?
Thank you. It's a very interesting technique. I'm wondering, because the spectrum you're using, it looks like it's about the same as the spectrum for laser hair removal, which targets melanin. So is there a risk of skin burning in darker pigments or just dark hairs on the skin?
Yes, so this influences our signal a lot. So patients, we use a razor on their legs to remove the hair, for example, before examination. But we actually looked into this with another study. It's not muscle-based, but we looked detailed into skin itself and whether, how the melanin influences the signal itself. There's actually a paper coming up that's published in a few months, I believe. And we try to, again, use fluence correction to correct for the differences in that. But that's a very, very important issue to raise. And also this is some of the difficulties we actually have when we use a pulse oximetry, where we also look into the detail whether maybe optoacoustic can kind of influence the issue we have with pulse oximetry. Yes.
Sorry, we actually have a question online, and I managed to figure out how this works. So I'm going to ask you the question. I think I also sent it to you, but I don't know if you see it. So there's a question, and someone who says, thank you for a great talk. If I understood correctly, the method is based on near-infrared spectroscopy, but with visualization. What are the limitations, except depth and reproducibility, like hair-making gas layer on the skin? Is some gel used? That's
yes, we use gel, okay. And it's a transparent gel. So not the ones we regularly use for ultrasound imaging. Does this answer the question? Not sure.
I don't know. Yeah, so the last one is for Crystal. Sorry.
So nice talk, thank you very much. I was just wondering, because you do this technique in children, how will you deal with growth, with weight gain? Because in longitudinal studies, those children grow. How will you deal with that?
Yeah, that's a very important question too. Again, I think reflecting on the question Professor Diaz-Manera asked, how are you able to do longitudinal studies and compare the results? No. So again, it's very the main limitation when you do the image acquisition is the penetration depth. So when they gain weight very, very fast, then it's going to change the signal values. We always, when we do the image acquisition, we can see at what depth we place the ROI, so the region of interest, and so then we can compare with this. And for example, if we see there's like a large decrease in signal values and the ROI is placed below 1 centimeters in comparison to the one we did before, then we know that's probably due to that. But when we compare the same patients and follow them up over a longer period of time, it would be important to place the ROI at the same imaging depth.
All right. Thank you, Lina, very much.
Thank you so
I think
much.
in the sake of time, we're going to move forward. And the next speaker selected oral presentation is from Eriqui Caldas de Almeida Araujo. No, if I could pronounce that correct. And it's about non-invasive assessment of histological changes in dystrophic and developing skeletal muscle in GRMD and control dogs using a bicomponent T2 relaxometry mapping.
Thank you very much. Good morning. Thank you for this opportunity. So, in very simple terms, when a muscle is injured, an inflammatory process rapidly takes place, characterized by vasodilation, promoting extravasation of plasma and macrophages into the injured zone. Then the tissue goes through a regeneration process, and usually within some weeks or months, depending on the extent of the lesion, the tissue is completely regenerated. In muscle diseases, however, the pathological condition induces chronic tissue injury, leading to chronic inflammation and incomplete tissue regeneration. And with time, this leads to long-term structural changes characterized by strong variability of fiber size, progressive muscle fiber replacement by fat, and the accumulation of connective tissue in the extracellular matrix, namely fibrosis. I like to distinguish changes in tissue composition, like fatty replacement, from changes in what I call changes in tissue water compartmentation that reflect rather the pathological alterations like inflammation, necrosis, and regeneration, and also the long-term changes like variable fiber diameter and expanded interstitial space due to fibrosis. So currently, the main image biomarker in the field of neuromuscular disorders is fat fraction. It allows precisely quantification of the rate of muscle waste and replacement by fat, providing an accurate picture of the disease severity and progression. On the other hand, pathological processes that precede fatty replacement can be detected using water T2, water T1, and diffusion tensor imaging metrics. However, despite of their sensitivity, we still don't know exactly how the different underlying pathophysiological processes and structural tissue changes impact on these different metrics. So there is today an urgent need for new biomarkers allowing to quantify specific pathophysiological changes that precede fatty replacement. So in this quest, I've been exploring the water T2 relaxation, first because this parameter is extremely sensitive to the biophysical environment of the tissue, and second, and very important, the water T2 relaxation in muscle is multi-exponential, and there are now several evidences that indicate that this behavior reflects the tissue water compartmentation at an histological level. So an evident working hypothesis is, can we explore tissue water compartmentation using T2 relaxometry in order to assess specific histological features? Knowing that a similar approach has already provided new important histologically validated biomarkers in brain applications. So in this study, the aim was to investigate the sensitivity and specificity of bicomponent water T2 mapping metrics to histological changes in the developing healthy and dystrophic skeletal muscle. In this MRI study, we have included 12 golden retriever dogs, of which 8 GRMD, which is the canine model of the Duchenne muscular dystrophy, and 4 age-matched controls. So dogs' hind limbs were longitudinally examined around 2, 6, and 12 months of age. The MRI protocol included a 3D gradient echo sequence for fat fraction mapping and a 2D multi-spin echo sequence for bicomponent water-to-2 mapping. This last one was accomplished by adjusting a bicomponent EPG signal model to the signal evolution within each voxel. In a different and similar cohort, we took muscle biopsies on the tibialis cranialis muscle in dogs at 2, 6, and 12 months of age. And histological analyses were performed on H&E stained cuts. Then we compared results between GRMD and control and evaluated their progression with age. So regarding MRI results, on the right, you have examples of the main components T2 map and the long T2 fraction maps of a control and GRMD dog at different ages. So our results revealed that fat fraction was similarly low in both groups, and the T2 of the main component and the relative size of the long T2 component was systematically increased in GRMD at all ages. We also observed a significant decrease of the long T2 value in younger GRMD dogs. Regarding progression with age, we observed a similar and progressive decrease of main components T2 in both groups, and a significant decrease of long T2 fraction in both groups, and this decrease was significantly more pronounced in the GRMD. So here are examples of histological images in these dogs, and we observed the absence of deposed tissue infiltrates in both groups, the presence of necrotic and inflammatory events in GRMD, and increased endomesial connective tissue space in GRMD at 12 months of age. Regarding progression with age, we saw a clear progressive increase in fiber size with following tissue growth, and a progressive decrease in the pathological events in GRMD. So the relations between our MRI metrics and histological observations were quite interesting. The similar progressive decrease of the main component's T2 value in both groups probably reflects tissue growth, characterized by progressive increase in fiber diameter and a consequent progressive decrease of the relative extracellular space. The systematically elevated T2 observed in GRMD can only reflect the pathological features which were exclusively observed in the GRMD dogs. The progressive decrease of the long T2 fraction with age in both groups must also reflect to some extent the tissue growth, while the systematically elevated values observed in GRMD must reflect disease activity as well. Interestingly, for this metric, the decrease observed in GRMD was significantly more pronounced, which could be explained by the progressive decrease of the pathological features which were exclusively observed in the GRMD. So it's quite remarkable that parenchyma vascular compartmentation model that has been previously proposed by previous studies and different groups provides a rather simple framework for interpreting these results. So in such a model, the main component represents primarily the parenchyma, while the long T2 component represents the vascular space, which does not comprise only the capillary network, but also all the microvasculature that is not resolvable in MRI. So from this framework, the progressive decrease of the parenchymal T2 with tissue growth in healthy dogs can be explained by the progressive decrease of the relative interstitial space, which should be characterized by a longer T2 due to its lower protein concentration. And the higher parenchymal T2 observed in GRMD can be explained by pathology-induced increases in the interstitial space and in membrane permeability. While the increased vascular space observed in GRMD and the steeper decrease of this metric observed in the same group could be explained by chronic inflammation and progressive decrease of the disease activity during the first year of age. So to conclude, bicomponent T2-relaxometry metrics provided sensitivity to microstructural remodeling during tissue growth, allowing to distinguish diseased from healthy tissue at all ages. It also seems to provide complementary metrics for tissue microstructure, as reflected by the distinct longitudinal trajectories of these metrics in GRMD and control. These results align well with the parenchymal vascular compartmentation model, providing a simple and yet physiologically meaningful interpretation of these results. Taken together, these results support the feasibility of assessing more specific histopathological changes in the tissue non-invasively, exploring tissue water compartmentation, for example, using multi-component water tissue relaxometry. So I think the biggest difficulty for translating this to clinics is the bias from fat signals, since in patients, fatty replacement is a really important feature of most neuromuscular diseases. Thank you.
Thank you very much. I think we have time for one question. Is there any question from the audience? Yes, you're in.
Thanks, great talk. What's the here you've used a slightly different model to in your EPG model to the one that's been adopted more widely. You've allowed more parameters to be free here in measuring these different components of T2, and you're still taking 17 echo, 17 data points. I mean, have you looked at the inherent stability of those fits and the uncertainty in the T2 components you're reading?
Yes, nice question. Yes, actually I'm not fitting much more than we already do. What I do here is that I'm using a bicomponent, so I have basically, because we've been using bicomponent fits, I would say, but we are trying to separate fat signal from water signal usually, and here I'm just separating two components of water signal, which is rather simpler, I would say, than separating from fat signal. And so the number of components is rather 4. So not much, actually, not many things has changed.
So to be able to get you to the coffee break on time, we're going to stop the questions now, so Eddie will be around, of course. And I would like to introduce the next speaker, who's Melissa Hoijmans. And her talk is entitled Conventional and In-Magnet Cardiopulmonary Exercise Testing of Patients with Neuromuscular Disease to Investigate Peripheral Causes of Exercise Intolerance.
Yeah, thank you for the nice introduction and the opportunity to share our data here today. Many patients with neuromuscular disease experience exercise intolerance, which significantly affects their daily life activities. The peripheral causes of this exercise intolerance is, in many cases, poorly understood, and specifically, I mean with peripheral causes, the mitochondrial and microvascular function. Clarification of these processes is essential to develop targeted interventions, maybe dietary, maybe exercise, maybe even therapeutic. In order to be able to measure this, you would like to measure this simultaneously, and we can do that by using an interleaved dynamic proton MRI and phosphorus spectroscopy acquisition to simultaneously assess microvascular and mitochondrial function. And often this is done in isolated small muscle mass exercise. And in this study, we're trying to incorporate that in an in-magnet exercise platform where participants can do a maximal leg cycling test in the MR scanner. And the goal was to investigate the feasibility of using this interleaved acquisition with this in-magnet exercise platform to investigate microvascular and mitochondrial function in patients with neuromuscular disease. And we investigated 2 separate neuromuscular diseases, both characterized by a certain level of mitochondrial involvement. And the first disease is chronic progressive external ophthalmoplegia, or in short, CPEO. It's a mitochondrial myopathy caused by the lesions in mitochondrial DNA, which play a key role in the oxidative phosphorylation. The disease is characterized by chronic bilateral ophthalmoplegia, which means like the hanging eye, so it's very clear from a visual assessment if you see these patients. And later on in the disease, also other muscles get affected. And the second disease we investigated is inclusion body myositis, which is a most common acquired myopathy after the age of 50. And this disease is characterized by progressive muscle weakness, most often asymmetrical, and often the quadriceps and the finger flexors are involved in the first stages. And besides inflammation, you see rimmed vacuoles, atrophic fibers, and also mitochondrial changes in biopsies. So in this study, we collected not only data in patients with neuromuscular disease, but we also obtained MRI data in healthy older individuals. So all NMD patients underwent a cardiopulmonary exercise test outside the MRI scanner to measure the level of exercise intolerance. And 2 weeks later, they also performed an in-magnet, all-out exercise test. And during that exercise test, we obtained interleaved proton MRI and phosphorus spectroscopy data on a 3 Tesla system in rest, during cycling, but also during the recovery phase. To give you an idea on how the exercise setup looks like. So this is a backside view of the MRI scanner in Amsterdam. And you see the MR-compatible ergometer. And the patients performed the exercise on this bike. And you can also very clearly see the biking pedals. And each of the participants were fitted with individual race bike shoes, and they were clipped into these biking pedals to be able to perform the exercise. We used a double-tuned phosphorus proton coil placed on top of the vastus lateralis muscle, and the participants performed an incremental exercise test until exhaustion, and we had a dynamic time of 4 seconds to obtain a proton image and a phosphorus spectrum. And the total duration of the acquisition was around 27 minutes. In addition to that, we also obtained a feasibility questionnaire post-exercise to get some idea on how well the exercise was for the participants to perform, because it remains in the MR scanner, which might be not so comfortable compared to outside of the MR scanner. To give you some more idea on how the data looks like, so we, to characterize the microvascular response post-exercise, we characterized the hyperemic effect, and we did that using multi-echo gradient echo images. And from those images, we used a mono-exponential fit to calculate R2 star maps, and then we normalized these R2 star maps in the vastus lateralis muscle per ROI, per time point, and characterized the time effect, which is now plotted on the right side of the screen. And we primarily focused on the post-exercise part of the curve, so it's behind the gray part, and we characterized the response post-exercise to get a surrogate measure of microvascular function. In addition to that, we looked into the phosphorus spectra. So here a typical phosphorus spectrum is shown on the left side of the screen, and we looked at the individual high-energy phosphate metabolites, and the amplitude below the peak is an indication of how much of that metabolite is present. And we also measured this over time, and we looked at the dynamics of the individual peaks. And by measuring the distance between PI and phosphocreatine, it's possible to measure also pH within the muscle. So that's one other measure we obtained. So just to summarize what key outcome measures we used in this study. So from the CPET data outside the MR scanner, we obtained a VO2 peak, and we showed that as a predicted maximum to get an indication on the level of exercise intolerance in these patients. In addition to that, we looked into the phosphorus outcome measures. So we determined phosphocreatine depletion levels, we determined end exercise pH, and we determined 95% PCR recovery times, which is a surrogate measure of mitochondrial function. And for the R2 star measures, we indeed characterized the post-exercise response. So we determined the maximum response, the time to peak, and also the slope. To dive into the results. So we assessed the feasibility using the questionnaire. So overall, all participants were able to perform the exercise outside the MRI scanner. 31 out of the 33 participants were able to perform it inside the MR scanner. 1 stopped due to cramping of the hamstring muscles, and 1 wasn't able to perform it due to contractures of the knee and ankles. Overall, we thought it went quite well, and we got some suggestions from participants to even improve the platform, because now it's a little bit limited in height. So if you're too short, it's impossible to cycle. But if you're too tall, it's also impossible to cycle, for instance. Now, when we looked into the CPET test outside the MRI scanner, we saw that each of the patients with neuromuscular disease had a certain level of exercise intolerance. So on the left side, in orange, you see the IBM patients, and in purple, you see the CPO patients. Now, then we used the MR data to look a little bit more into depth. So I'm showing you the phosphorus data. So on the left side, you see phosphocreatine depletion for the 3 different groups. In the middle, end-exercise pH. And on the right side, 95% PCR recovery times. And if you look closely, you can very clearly see that there's no differences in PCR depletion between groups, which suggests a similar level of exercise intensity during the test. We don't see group level differences for end-exercise pH, but you do see some variation in IBM and CPO patients, and it tends to be a little lower. And if you look at 95% PCR recovery times, you can very clearly see that these values are prolonged in the CPO patients. We do see some variation in both the healthy controls and the IBM patients, but no clear differences there. And then when we look into the R2-star outcome measures, on the left side, the maximum hyperemic response, in the middle, the time to peak, and on the right side, the slope. You can clearly see that there is some variation between individuals, but on a group level basis, we don't see any differences in the R2-star outcome measures. So overall, we thought that the feasibility questionnaire showed promising results in these 2 patient populations, so we're planning to expand it to a wider range of other patient populations there. Based on the CPET results, we saw that there's a certain level of exercise intolerance in all these individual NMD patients. And then we dove a little bit deeper and explored the peripheral causes of these limitations on exercise level in these patients. And we saw that there is a significant difference in 95% PCR recovery time, which suggests that there is some mitochondrial dysfunction going on in the CPO patients. We didn't see such a clear message for the IBM patients. And when we compare that to findings in literature, we saw that the findings for CPO align really, really well. But when we look into the findings for IBM, it was a little less clear, and most of the results are based on biopsy results, where there are also a lot of variability present in these patients. But overall, we believe that this interleaved approach in combination with the MR exercise ergometer to do an in-magnet test is very promising to explore the peripheral causes of exercise intolerance. And that was the talk. Thank you. If there are any questions, happy to take them.
Yeah, we have time for one question. Pierre, go on.
Yeah, thank you, Melissa. It's great and reassuring that multinuclear interleave NMR is alive. Thank you. Thank you very much. I still have a question. I understand that measuring the BALD signal is much easier than getting the ASL signal. However, as you know, when you measure BALD is not a measure of perfusion. It's influenced by many other things. And I also know that, well, trying to do ASL in the thigh is a challenge. But are you thinking about that? Are you working on that? Or is it a goal for future investigation?
I have thought about it. I'm not currently working on it, but I agree it would be ideal to do it with ASL instead of the R2-star maps we obtained here, because you have a more clear message if you would use that. But I think ASL is very tricky, and especially in rest, to normalize it to certain values, because resting perfusion is so low in muscle. But yeah, ideally, I would love to. But yeah, we're working with Philips, and we're not able to do interleaved anymore, so I'm not working on this topic. So maybe Siemens could bring this a little further in the future.
All right, thank you, Melissa, very much. I think we have to move forward. The next speaker is Puneet Bagga, talking about mapping skeletal muscle mitochondrial oxidative phosphorylation in health and SMA using a novel technique, OXCEST MRI.
Thank you so much for kind introduction, and I appreciate the opportunity to present our work. I'm a assistant member in radiology department at the St. Jude Children's Research Hospital. We work with a lot of pediatric patients and also survivors of cancer who show up with some sort of mitochondrial impairments, both in the brain and muscle. But I'll be talking about muscle here. And I'll be talking about muscle in the context of spinal muscular atrophy and a technology that we have been developing in parallel to the multinuclear spectroscopy that was so eloquently explained in the previous talk. So just a brief background about SMA. It's a motor neuron disease caused by a loss caused by the mutation in the SMN protein, SMN1 protein, and it leads to motor neuron degeneration and then neurogenic atrophy in the spinal muscular atrophy patients. Interestingly, 10 years ago, it was shown in the muscle biopsies of these patients that the mitochondrial DNA copy number is lower, and it's also associated with the severity of the disease. So type 2 will have lower mitochondrial DNA copy numbers than the type 3, and they'll all have lower mitochondrial DNA copy numbers than the controls. So that led to an interesting paper from the same group showing impaired mitochondrial OXPHOS, or not impaired, but remodeled mitochondrial OXPHOS activity in the upper limb muscle groups, here as shown with the 31 PMR spectroscopy. So in the SMA patients, it was shown that the biceps were not impaired compared to the triceps, but it was shown that the PCI recovery time was somewhat lower, but also the white muscles or the fast twitch muscles basically remodeled to the red muscles. And yesterday, Felix Kogan presented a talk on creatine cyst. So this is the technology that we have been also working on to show the reproducibility of. And we showed that the creatine cyst is pretty reproducible in lateral and medial gastroc muscles, but not so much in soleus muscles. But also we showed that it's a very moderate sort of reproducibility in the lateral and medial gastroc muscles. So we came up with a newer technology that, excuse me. I'm sorry. It's okay. So we came up with a newer technology called OXCYST MRI. It basically, thank you. It basically saturates creatine amine protons at 1.8 and minus 1.8 ppm. And if I can play the video. Here. So post-exercise, as we've seen in the other talks as well, the phosphocreatine is depleted and creatine is accumulated. So what happens here is when you do the plantar flexion exercise in a healthy subject, there's an accumulation of creatine, and it recovers to rephosphorate the phosphocreatine in an exponential measure. So what we have seen here is that lateral gastroc and medial gastroc have a pretty high reproducibility or exponential curve reproducibility, whereas in the soleus it's totally dependent on the activation, muscle activation. So next, I'll just talk about the experimental setup here. So we do OXCYST, as explained yesterday again by Dr. Kogan. We do a reference image, we do a B1 map, FOSSER, and then we do a pre-exercise test. Takes about 2 minutes. And then we do exercise for 2 minutes, plantar flexions. And then we do a post-exercise test recovery for 10 minutes. And then we do a FOSSER for B0 mapping, B1 mapping, and then reference. We also do 31 PMR spectroscopy. Rest for 5 minutes, 2 minutes exercise, recovery for 10 minutes. This is our setup right here. Okay, so OXCYST showed a very nice association or correlation with 31 PMR spectroscopy, because we saw that in lateral gastroc and as well as the medial gastroc, the recovery was very highly exponential compared to phosphocreatine, as you can see here in the third figure. And we did it in 7 subjects, and the correlation is pretty high with the 31 PMR spectroscopy, thereby validating our technology. I also want to mention here that we are able to generate a pixel-wise map of phosphocreatine recovery here, as we can see in this figure. Next, we tested the reproducibility of our technology by rescanning same subject twice. We saw, again, pretty high reproducibility of lateral and medial gastroc, whereas Soleus was somewhat reproducible. All right, so moving on to SMA, we saw that here's a case from a 22-year-old male who is a type 3 SMA, and we see that there's a lot of fatty infiltration in this muscle group, in the calf muscle, and we did the same exercise, plantar flexion exercise. We saw a recovery of creatine over time. We measured the tau. And we saw that compared to one healthy age-matched control, the SMA survivor had higher or longer creatine recovery time constants, thereby telling us that there's an OXPHOS impairment in this patient. So next is an example of an 8-year-old or 9-year-old subject who was diagnosed or predisposed to be a type 2 SMA patient. But he started with nusinersen. It's a therapy that is given intrathecally, and it increases the SMN2 expression, thereby increases the, it recovers the motor neuron function, it seems. And so we see that the recovery for the lateral and medial gastroc post-exercise in this patient is pretty low. LG has a little higher component here, as you can see, but it's because it's a very noisy data, probably because of motion. As was noted in the previous talk, and by many others as well, is that lower limb muscles, especially in SMA patients, are more impaired in the more aggressive form of SMA. So what we have been trying to do is implement OXYST, this technology, in upper arm scenario. So we're using a flex coil, as you can see here, on the biceps, and then we pull a band, a resistant band, to engage the biceps brachii muscle, and we again perform the same experimental setup, do a pre-scan cyst, pre-scan cyst, and then pre-exercise cyst, exercise, and then post-exercise recovery cyst, and we see that in the biceps muscle, as you can see, over time, the creatine is recovering, whereas there's also, we can see the engagement of the triceps lateral heads as well. Evidently also we saw that the tau CR, the rate of recovery of creatine, is somewhat associated with age, which we are yet to see in the lower leg muscles. We will be implementing this technology in the upcoming scans with the SMA patients and the other patients as well. So to conclude, we think we have developed an OXPHOS-based imaging method which shows a good validity or good agreement with the 31-PMR spectroscopy. It demonstrates good repeatability between scans in healthy subjects. It detects OXPHOS deficiency in individuals with SMA, as I have shown, and it's also feasible for the upper arm imaging, or at least we are trying to make it more feasible for upper arm imaging. In the ISO, I'm sorry, I did not mention, but we did isometric exercise for the upper arm, and we did plantar flexion isokinetic for the lower limb calf muscles. Okay. And then with this, I would like to thank you, and if you have any questions, I'll be able to answer. Thank you.
Thank you very much. There's time for one very quick question. Pierre.
Yeah, well, thank you very much. It's probably a very naive question, but if I read properly, you creatine recovery curve in normal, the time constant is about 45 milliseconds, while in young children, the creatine rephosphorylation time constant is rather 20-25 seconds. So how is it possible? How do you explain that?
The creatine recovery time, it depends on the muscle as well.
Same muscle.
For the same muscle, I wouldn't know. I mean, there's definitely a engagement issue that was discussed yesterday, but again, I wouldn't know why a young kid would have a lower. We know that with age, there's an increase in the tau CR or tau PCR. That's been shown by many groups, especially at Johns Hopkins. But if you're talking about the SMA patient here, he had a longer tau CR.
Okay, maybe we can move that discussion to the coffee break, and now get to the very last but not least, a presentation of the session. It's from Michele Giovanni Croce, talking about magnetization transfer imaging in late onset Pompe disease.
Thank you for the introduction, and good morning, everyone. I'm Michele Giovanni Croce from the University of Pavia, neurology resident, and today I'm talking about magnetization transfer imaging in late onset Pompe disease. So these are my disclosures. Magnetization transfer imaging is a relatively novel in the field of neuromuscular disease MRI technique that exploits the magnetization exchange between two proton pools, the free pool related to protons in free water, and the bound pool, the proton bound to macromolecules such as glycogen. The interaction between the two compartments can be measured by the exchange of energy from the bound to the free pool by applying a radiofrequency pulse known as the MT pulse, which produces the MT effect, which can be quantified as a magnetization transfer ratio. So we do know from previous studies that musculoskeletal tissue actually has a pronounced MT effect, and that can represent a sensitive measure of muscle damage in different conditions, may be related to progressive fiber loss or fat replacement. In Pompe disease, these actually results are much controversial because we don't know if the alteration in MTR are related to the accumulation of glycogen or fiber loss, and so it was of interest to study MTR in pre-symptomatic patient with not so relevant clinical alteration and not so relevant fatty replacement. This led to an interesting study from the Spanish group in 2021, which found actually that in 4 muscle of the thigh there was a good correlation with muscle function tests and MTR parameters, and also significantly lower MTR values in LOPD patients versus controls, with lower average MTR values in symptomatic versus controllers and in asymptomatic patient, and significantly lower values only in adductor magnus with muscle with not yet fat replaced were considered with the fat fraction less than 20%. So we decided to include genetically confirmed LOPD patient aged more than 10 years old, divided into 2 different groups, early stage patients asymptomatic or with minimally clinical involvement, and symptomatic patient with a broad range of symptoms and clinical involvement. We recruited 66 individuals, 32 LOPD patients divided in 15 early stage patients and 17 symptomatic patients, and 34 age and sex matched healthy controls. Patients underwent comprehensive clinical evaluation with manual muscle testing and the currently used clinical test, and also patient-reported outcomes, and 30 MRI scans of the thigh with the calculation of the fat fraction with the 6-point Dixon method, in particular 3 slices of the thigh, a medial, a proximal, and a distal one. We drawn actually 11 ROIs corresponding to 11 thigh muscles with Daphne software, and then a subsequent validation by an expert operator, and then the computation of the fat fraction with the FATTI-Rayod algorithm, and the calculation of MTR with the formula showed in the slide, which was then extracted from each slice position and averaged over all slice position. So these are the clinical assessments, which of course are very different from early stage to symptomatic patient, and also to the pulmonary evaluation and to the patient-reported outcomes. First of all, we can say that there are significant differences, and in particular significantly lower MTR values in all the patient compared to controls. This is true for the mean MTR values of the thigh, but also for most of the muscle studied in the thigh, except for rectus femoris and sartorius. When the three groups were analyzed separately, there were no significant differences in the mean MTR of the thigh between controls and early stage patient, although there were differences between controls and early stage, between controls and symptomatics and between early stage and symptomatic patient. But when muscle by muscle comparisons were made, we did find actually a significant difference in adductor magnus and gracilis between controls and early stage patient, with significantly lower values in patient compared to control. We then wanted to see if there were differences in MTR parameters in muscle not yet fat replaced, and we decided to choose a very low percentage of fat replacement, 10%. We didn't find a difference in terms of the mean MTR values of the thigh between patients and controls, but when single muscle comparisons were made, we actually did find a difference in three muscles, gracilis, adductor magnus, and biceps femoris short head. And when we compared the three groups, we did not find a difference between controls and early stage in the mean MTR of the muscle, but again, adductor magnus and gracilis showed a difference in the MTR values, with lower MTR values in patient compared to controls. We also found a negative correlation between MTR and the average fat fraction of the thigh, and also a significant correlation between MTR and most of the clinical tests conducted on those patients. So we can say that MTR effectively distinguishes symptomatic patients from healthy controls with lower MTR values in patients. It does not seem to distinguish early stage patients from healthy controls when we consider the mean of all muscle, but when we do single muscle comparison, we were able to find significantly lower MTR values in adductor magnus and gracilis in early stage patients versus controls. And we also found similar results when the analysis was performed in muscle not yet fat replaced, with the fat fraction, I'd say normal, with less than 10%. And also we found a good correlation with clinical tests and a negative correlation with fat fraction. So we can say that MTR seems to be altered before fat replacement has occurred, and as hypothesized in the previous study, its lower values are probably related to an early loss of muscle fiber, so it is a probable tool to see a high sensitivity to muscle fiber loss and damage, and maybe to monitor early signs of progression and decide early timely initiation of therapy. Of course, there are some limitations to this study because we did not use a gold standard test to study the amount of glycogen, so we are not able today to actually verify the premises of this study, invasive or not invasive gold standard study to study the glycogen, of course. And we also need a longitudinal follow-up to verify the decrease in MTR and also the subsequent fat replacement. So I want to thank all the people who took part to this study, starting from Professor Picchecchio, who is here in this room, and also I want to thank you for the attention.
Thank you. Are there questions from the audience? Oh, are you in need of coffee? I have one. Good. I prepared many and I could only ask one.
Yeah.
So I was wondering, because I think it was also mentioned earlier this morning, that Pompe disease patients also have like increased T2 signal intensity, so there's more water.
Yeah.
So do you think that your MTR results reflect this increased water, or does it show something else?
I mean, maybe we don't know what MTR represents, but I mean, as I said in the beginning, MTR seems to be represents the exchange between the bound pool of protons to macromolecules and the free pool to water. So maybe it could represent, protons related to water, but in other conditions such as IBM or SMA or also LGMD, previous studies actually found lower MTR values. So maybe, which was also our result, so maybe it is more probably related to an early loss of muscle fiber than an accumulation of water as we see in water T2, muscle water T2 alteration. So, I mean, that's my guess, but I don't know.
Thank you. I asked for a guess.
Yeah.
Well, thank you.
Thank you.
So with that talk, that closes the session, if there are no more questions. Oh, sorry, there was one. Go ahead. Yeah, if you press it, then the light goes on, and then it should work. Yeah.
How did you do MTR? Is it easy to do in a multi-center trial? Is it something that vendors have already made now, or not yet?
I think that it is quite easy to perform it in a multi-center setting, yeah, and because it is actually fast to perform, and so it would be easy to apply it in a multi-center setting, yeah.
Using any of the vendors?
I'm sorry.
Using any of the vendors of the MRI vendors, Siemens, Philips, GE, doesn't matter.
Well, I can support, as Michele is a neurologist, so as radiologist, we have a Siemens machine, but it can be applied also to the Philips.
Okay.
And it's quite quick and quite easy also to set up compared to other techniques in a clinical setting.
Thank you.
Thank you.